we're happy to have Karsten Buschard with us today to discuss, fenofibrate and its role in a patient who was diagnosed with type 1 diabetes. In the paper they submitted was in August 2019, and we're coming up on 2 years now. So it's really interesting to see where that story went. So thank you and welcome, Karsten, for joining us. Take it away.
Yes, thank you for the invitation. I'm very pleased to tell about our studies. It started actually back in 1990, where we discovered that sulfatide could play a role in type 1 diabetes. And sulfatide belongs to a group of molecules that is called sphingolipids. It was discovered for more than 100 years ago, and people do not know what it is, and therefore they named it after a sphinx, so it's called sphingolipids. And it's highly interesting molecules. They consist of a sugar group. Here it's galactose for sulfatide, but it could be glucose, and there could even be two sugars. Then there's a backbone of serine, an amino acid, and you can see the nitrogen atom here. So a serine is present, and then there are two fat tails. So it is a fat molecule, but very specially has an amino acid and a sugar group, and for sulfatide, a sulfate group. And these are sphingolipids. They are mostly present in the brain and neural tissue, but also in the beta cells that in many ways resemble neural cells. Here you can see a human beta cell, and there are secretory granules with insulin. You can see one come out here, and this has just been empty. And that we have stained with a sulfatide antibody, and you can see the gold granules that is staining for sulfatide in these secretory granules where there's a lot of insulin. And what does it do there? Here we have taken an example in laboratory and made insulin crystals. And insulin is stored in the beta cells as crystals. And you can see a nice crystal here. And this is bigger magnification. And this crystal can last for 2 to 3 weeks when sulfatide is present, like here. But when there's no sulfatide, the insulin crystal is deteriorated within 2 days. So sulfatide plays a physiological role for preserving the insulin crystals, and we have stored insulin in that way for 2 to 3 weeks. It must be in crystal form, otherwise it would fibrillate. It's also helped by secreting insulin and folding insulin, so it's very important physiologically for the beta cells. Then we have found that this compound fenofibrate is important, and fenofibrate has been used for decades to lowering cholesterol. It might not be very good, and to that, and it has been surpassed mostly by statins, but it has other interesting features. Actually, it enhances the sulfatide amount in the islets of Langerhans. When we are feeding mice with fenofibrate, it has almost double as much sulfatide than the control mice. And highly interesting, as I told you, there was both sulfatide with low fat chains and with high fat chains, and the low fat chains sulfatide are making all the physiological have all the physiological properties in the beta cells, as I just have shown you an example of. But the long chains sulfatide are anti-inflammatory. These are the anti-inflammatory molecules. And you can see that behind my picture, or your picture, it's nearly double up, or more than double up the amount of sulfatide when giving fenofibrate. And that is very interesting. It does not harm or anything with the physiological role of sulfatide, but it more than double up behind our pictures the amount of the anti-inflammatory sulfatide. And then we have used it in nut mice, and nut mice are a highly interesting animal model for type 1 diabetes. I'm sure that Bart will agree on that. And you can see within 200 days, 2/3 of them, about that it vary a bit, develop type 1 diabetes. But if you gave these mice fenofibrate and we gave it in their diet, then none of them developed type 1 diabetes, really none of them. And that is very like a miracle, so to speak. Usually there are difference on the two curves, but here none of the treated mice developed type 1 diabetes.
Karsten, was this prevention or intervention?
It was, yeah, we gave it from before the diabetes. I think we started here at the...
Okay, so prevention.
Yeah, sure.
What about intervention?
Yeah, we have made that, and then half of the animals, of these animals, got out of diabetes when having fenofibrate. So half of these were cured when giving them fenofibrate. Then we were lucky to get in touch with a patient. It was a 19-year-old girl that have developed type 1 diabetes classically with high blood glucose. You can see at this y-axis here in red, you can see the blood glucose. These are the insulin dose, so you might not see them from the very beginning. But she had up to 30 millimole, that's about 150, 200 in American terms. So high blood glucose, a bit of ketonuria, and she was antibody positive for GAD antibodies and IA2 antibodies. So classically, a classical type 1 diabetes. And then within a week after she was diagnosed, she started on fenofibrate. The insulin dose was quite high, but after 18 days, she came out of insulin. Here she was on a backpacker tour to Sri Lanka, and the blood glucose was a little bit high. You can see on the other y-axis blood glucose. And she took a small insulin dose only once because she had an infection. But ever since, she has been out of insulin now for nearly 3 years. And actually at the last 2 tests, her IA2 antibody levels are decreasing. The GADs are not, they used to be fairly constant. And she has C-peptide in the lower end of the normal value. So for her, it's a miracle. And whatever it turns out to be in the future, she has at least 3 years without insulin, and she have a normal life. Then we have started a trial in Copenhagen. It should be a double blind as good as it can be, and it is with 58 patients plus and minus insulin, and we have more than half enrolled, and they have good compliance. They have no side effects at all. As I told you, it is a very used and safe product, this fenofibrate, and we expect to have a result within or 1.5 years from now. I should mention maybe my last slide, that is a table of gene variants. You know, we have different blood groups, blood types, and there are also in these enzymes that make sulfatide or involved in sphingolipid metabolism. There are also some gene variants, and when they are connected to, and they are connected to type 1 diabetes. So people with type 1 diabetes, for example, for this one, they have a 47% higher risk of getting diabetes. It's not of any value really for the individual patient, but when we have found these 8, it tells us that there could be or must be something involved in the sphingolipid metabolism in the type 1 diabetes pathogenesis. So it's theoretically highly interesting and support that there could be something with sphingolipids in type 1 diabetes. There's nothing here in type 2 diabetes, I can see. Yeah, maybe I should stop here or...
No, this is great. This is really interesting. I would invite some questions from the audience. Anyone have a question for Karsten?
Maybe I can expand a little bit, because it's a very complicated story, but it started off with a Lancet paper many years ago in early 90s of Karsten, where he showed that patients have autoantibodies against sphingolipids. So it is actually also involved in the pathogenesis. And as Karsten was indicating, it depends on the length of the chains, the fat chains, whether it's protective or susceptible. But what is really interesting is we showed with Karsten that if you have these long tail sulfatides, they actually inhibit the T cell response to insulin. So they are really anti-inflammatory. So in a nutshell, what Karsten has shown is if you take fenofibrate, you actually increase the good sulfatides, which are protective of the beta cell. And this is very much in line with our idea that if you give beta cells some peace of mind, the immune system is no longer interested in that. So that is in a nutshell what Karsten has shown. It's even more complicated, but I was glad that Karsten gave you the high level introduction. But this is what this has been doing.
Yeah, I think the high level introduction is important because as we've talked about before, Bart, we're trying to engage all members of the scientific community that study type 1. So, you know, from the patch clamping people to the GWAS people and so forth. So it is good to have sort of a real overview. I would ask, you know, I'm very curious about this, you know, the study that's going on in Copenhagen. Can you give us a little more about that, Karsten? Is there any more to talk about in that study?
Yeah, we have patients newly diagnosed as soon as possible after the diagnosis, that could typically be within 1 week or at the most 2 weeks. And they are in the age from 16 and upwards to, I think, 30 or 35. Typically, they are in their 20s. And all of them say yes to participate, and then they would have received placebo or fenofibrate. They do not know, of course, and we do not know either. And it's going well. They have no side effects, and we are all very excited to hear about the results, which would be within 1.5 years. I should say that these patients do not get anything else than fenofibrate, but our patient here were also low in intake of refined carbohydrate, so she did not stress her beta cells, and she was also easy with gluten, which also might support the effect. But he have taken this 160 milligram of fenofibrate every day, every evening. She takes it, and she's doing well, and it started her studies and so on. So it has really been a pleasure to follow him.
Have you noticed if a patient doesn't take their medication for a day, does anything happen? Like if you forget or something, is that an issue?
We do not know, and she said that he's taking Fenofibrate every day. I don't know whether she has forgot it once, but
Mhm.
But this is but Rachel, this is a medication that is like the pills you take against the high blood pressure, right? So if you skip a day,
so high cholesterol.
it's building up. Yeah, but I'm trying to say it's a more chronic effect. It's not like the anti-conception pill that if you forget it, you can get pregnant. This is something that it doesn't really matter that much if you skip a pill.
Got it. Thanks.
Have there been any, Bart, you made the distinction earlier between the, whether or not this was a prevention or, a reversion. And
yeah, this is a reversion. And of course, I would love to treat antibody positive pre-diabetic, type 1 diabetic persons before they have the clinical diagnosis. I would love to do that.
But Karsten, what I would love you to do is take patients that have had disease for 10 years and get them out of hibernation. Because if you give the islets comfort, if a very, very ancient hypothesis called beta cell rest is true, that you could actually cause that by this. And it's understood why you take new onsets, because it's more easier to measure. But the majority of patients are not in that group. And I would love you to take patients that are 10 years out.
Yeah, and I would love to do it as well, but then we must have some inhibition of the T cells so that waking up beta cells, so to speak, are not destroyed.
You and I showed that happens. But the point is that the inflammation is also going down as soon as people start taking insulin, right? We know that.
Yes,
so maybe that is already partly taken care of. And as you know, my contention is that immunotherapy alone will not be enough, nor will beta cell therapy. Now, if your insulin therapy gets your inflammation down and gives your beta cells rest, then on top of that, fenofibrate might even protect the beta cells.
Yeah, it could resemble a bit, as you know, in pregnancy, early pregnancy, there used to come or often come a certain amount of C-peptide suddenly, not in the late pregnancy, but in the early pregnancy.
Yeah.
So it's of course a possibility, and it could be highly interesting to do. I agree on that.
And as you know, in pregnancy, islets start producing epidermal growth factor, which is protective. So that is one way of making islets happy again and start producing insulin during pregnancy. But I kind of have a gut feeling that this sulfatide might do the same thing. That's what I'm saying.
Yeah, I agree with you. Yes.
How difficult is it to get one of these sort of short trials? And it seems pretty simple. I mean, how difficult is it to get one of these trials off the ground, Bart or Karsten?
It depends where you are.
Can be because all kind of permission should be obtained and money should be obtained, and then the patients and so on. So it has taken a year to start up for a PhD student, MD, to perform this. It's not something that is just done.
Well, Karsten, let's take it from a different perspective, because many of you are familiar with clinical trials. This is an over-the-counter drug, right? I mean, it is safe. It is already approved by FDA, EMA, and so forth. So the drug is the easiest part, and most of the time it's the drug that is causing most of the problems getting FDA involved. So it is very easy to get through. The problem is that it's also off patent, so there is no financial interest in the manufacturers of this drug. So it has to be coming from an academic source to run this trial. That is one problem. And the other problem is that if people know that you can buy this, it is not inconceivable that the placebos are not really placebos. But in the end of the day, you want to, everybody has the right to do. People can also start eating low carbs and low gluten. So that is not a problem. The other problem is that running a trial in the US compared to Denmark is a world of difference. And I can speak to this. I did a trial on toll disease, as you know, in Europe, and it took 5 people to do it, and we could start it in a year and a half. It took 45 people here and about 20 times the amount of money to do half a size of a trial in the US. So that has to do with all the regulatory issues, all the insurance issues. As you know, in Denmark, healthcare is free and people are automatically insured and protected. So those are the generic issues. So I think we in the US should be glad that Karsten is running this trial in Denmark, and there's no need to repeat that trial. What could be repeated is doing a trial, as Karsten says, before the clinical onset. But I really want things to be tested long after, because that's where the unmet needs are. And maybe you can take different endpoints. Maybe we can look at hypos. We can look at the time in range type of things. So Karsten wants to have the slam dunk home run. Karsten, these are American expressions.
Yeah, I know.
I know you know. But that means insulin independence. That is the holy grail. I think that if you can avoid hypoglycemia by restoring a baseline level of C-peptide 10 years after, during your college years, that is clinically meaningful, I would argue. And I would love to hear your opinion to that, Monica and Rachel and anybody. I mean, this is what we need now.
Yeah, I agree. I mean, my sister has type 1, and there's so many people I feel like who have type 1 and have had it for a long time, and there's all this really cool unique studies that are going on, but it's like, oh, I got diagnosed 10 years ago. That can't really help me. No. But you know
what, that is a travesty, that feeling, and it's we kind of to blame ourselves, and FDA gets the blame, because FDA only counts area under the curve of stimulated C-peptide, so in other words, beta cell function, as an endpoint. But patients don't really mind injecting insulin if it means that they won't get complications, they won't have hypoglycemia and so forth. That is not the worst bit, right? Mm-hmm. And taking more or less is not the worst thing either. So I think that we should have a lobby to also have, and with the toll DC trial, I told those patients that they had no benefit, and they were on average 12 years after diagnosis. Maybe pretty close to your sister, Rachel. I'm not sure. Is that 12 years ago?
She's at 20 years now.
Or 20. But the interesting thing was, although we told them don't expect anything, they are now all-time low in their HbA1c, 6.2, 6.5, right? So that is, and they're in the mid-20s, which is a difficult time to be in range. Right. Yeah. So I think that there is something to be gained, except not if the FDA is so simple-minded that they only will take stimulated C-peptides.
Have you had conversations with them about?
Well, I'm actually writing a paper for the Lancet as a kind of Don Quixote, which I call a call to action, where I want to say, listen. There has been a recent paper where we are staging the disease. So now if you have 2 antibodies, but not diabetes yet, it's actually diabetes. So in other words, that allows you, the FDA will allow you to put people in trial. I'm going to write, I want to have a different consensus, and that is, what do we call a cure? Or what do we call a clinical benefit? And clinical benefit goes in many different ways. You can stop disease progression. It doesn't mean you stop taking insulin, but it means that your beta cells are not destroyed. That is where Karsten is active in that arena, where I am active. But it also means preventing hypoglycemia. It means preventing complications. And then we have, of course, preventing dysglycemia, which is another measure, right? So to avoid, so that's a very early one, kind of a secondary prevention. So there is I identified at least seven areas where there is a clinical benefit. For example, relapse after eyelid transplantation or stem cell therapy. That's meaningful, right? We haven't even started that yet. We have so as an immunologist, and when I make people tolerant again, when you're no longer attacking your eyelids, for me that's a cure. I know that's not the same for a patient, but at least we take that box. Then the endocrinologists have to do their tricks to get the beta cells back to work. But I want people to be a little bit more perceptive to new ways, new other definitions of clinical success. And as you know, I work in a cancer center. 10 years ago, we would only, the people would, the oncologists would say, the removal of the cancer, that is a cure. Now they say, if it stops growing, it's a cure. And they even say, listen, it's a clinical benefit if I can extend your life with 5 months, which is something we've been doing in diabetes for a long time, right? So for way longer than that. So in cancer has been telling me that we have been putting the bar too high. And now I think we need a lobby and discussion going on that involves everybody, patients, their families, scientists, physicians, FDA, regulators, sponsors, pharma, that we all start to understand, listen, there is many bars that we can jump over. And the first one is one that Karsten is doing. Well, Karsten actually is lucky with this case. Forgive me saying that, Karsten, that this patient is insulin independent. Even if you stop progression, that is a miracle, right?
Yes,
that will ensure that you never get complications. So I think we should, start discussing and educate each other, including the FDA, that we have we have much more to offer to patients, and we're ignoring all those precious people that have had disease for 10, 20, 30 years. And I think they have insulin, they can make insulin. So anyway,
if I must say, hypos were mentioned, that fenofibrate seems to increase glucagon secretion and lower insulin secretion. So I would suggest, I would love to have a trial to see whether it can diminish the amount of hypos.
Yeah, Karsten, and that is the combination that the pump people, the closed loop folks, are trying, because that is the perfect storm to give eyelids rest, right? It's not just insulin; it is the yin and the yang.
Yes.
So I think that is why
glucagon.
That's why I would urge you to do a trial in established diabetes, so that we can and then use time in range and hypo and so forth as
hypos could be highly interesting. And if some of you are very hurt by hypos, you might could as a case start on this, because it is safe, and some people are really hurt on hypos.
Yeah.
Ever since a mother told me of an infant that she was putting her alarm 4 times a night to check on her kid, I realized that this is the first thing to do. Be sure that people don't get hypos and just wake up, and then we're back to all the other palliative care. But that, I think that is something that we need to focus on now.
Yeah, that's very important for the patients. It is. I would ask you guys, what about partnering with like Inish O'Doherty, who's at CPATH, to try to streamline some of these clinical trials and modify them in the ways you're talking about? I mean, that's their MO. CPATH is talking about designing trials that are streamlined, accelerated, and really are powered to get the,
Monica, you use the word power, and that is often the deal breaker, because we are pioneering something. Nobody knows anything about intervention after 2 years after diagnosis. Nobody knows anything. So we just don't know how to power it. And it's a possibility that you may have to put in 1,000 patients to make FDA happy, right? And that is going to cost tens of millions. So that is the problem. So I would prefer to have an academic trial where we get philanthropy to pay for some. And I actually showed this data to my dear friends, the Wanek family, last week, literally. And the one thing that Todd Wanek picked up, the CEO of Ashley, is the fenofibrate story. And I told Karsten, I said, Karsten, we need to talk about this because, and their kid has diabetes for 15 years.
I have a question regarding this trial. So I look at this picture and I see that it's quite substantial results after, say, 20 days. So what I don't understand is if this is an over-the-counter drug that has been shown to be safe, why have a clinical trial that is longer than, say, 50 days? It seems like if this is not a very exception from the majority of people using this drug, it feels like you would see crazy good results within 50 days. So why not have a trial that is only 50 days long? And I mean, if you do that with 50 people, safety is already proven, and you see, I mean, my daughter has type 1. She got diagnosed 8 months ago. If I could lower her need of insulin from 20 units to 5 units, which means that the hyper and hypo would go down substantially, that would be insanely valuable for me. I would pay all the money I have to get that.
Yeah, I think that it's important to start as soon as possible before the T cells really are having a point of no return. But I can agree with Bart, and it could be worse.
I don't think so, Karsten.
And please, and I think that your daughter's hypos could really benefit. And I do not have a slide here, but it's in one of our recent publications that we have found that it increases glucagon, fenofibrate increase glucagon and lower insulin. So it could.
But Karsten, you know that we published together that sulfatide is by itself anti-inflammatory.
Yes, yes.
So it has what I am advocating to do to both suppress the immune system, modulate the immune system, yes, and to make beta cells happy. So one molecule does both of this. So I disagree that you first should have the T cells or prevent. That's too late anyway. I think that all that matters is local, what happens locally. And if you can get these doses there, I think it should be done. And I agree with that. If it's a black and white of insulin in 20 days,
yeah,
that you should see very quickly.
Yes,
but the point is that, let's be fair, Henrik, it's unlikely that this will work the same way in everybody. Yeah? Karsten also discovered that low glucose can actually get people off insulin for up to 2 years, but many others did not accomplish complete insulin independence, but all of them are very low in their insulin needs. Right. So what you were saying, Henrik, is important. It's not how much whether you take insulin or not. It's whether you are let's say partially insulin independent, which gets rid of all the rough edges. So that is something that should be the expectation. Yeah.
Could that play into creating a fast track trial or no?
No, the problem is it will impair a fast track trial because people will start doing that, right? Mm-hmm. You can go to your physician and say, hey, I want this. Certainly in America, the patients have much more power to force physicians to prescribe, right? That's not the case in Denmark.
Yeah.
But here, my fear is that, and it's fear, quote unquote, because I wish anybody can give this a shot, right? If it doesn't help, at least it doesn't harm. So that is the problem with this trial as well as gluten-free, right? If you want to test whether gluten-free diet will put people in better glycemic control or remission, you cannot tell anybody to start eating gluten anymore, right? So and so that is the problem. It's like vitamin D. Those trials have been terrible because everybody goes to the store and gets their supplements, and good for them, but the trial is very difficult to be designed.
I may should come up with one other thing here at the very end. That is that serine is maybe a crucial molecule. We have a big brain, much too big than other animals, and we need a lot of serine to make all these sphingolipids, and serine might be low. So make sure that in your diet you eat a lot of serine, and that could be eggs and fish.
Well,
and
thanks
so
for introducing another confounder now, Karsten, but that makes it...
No, it's important. It is.
I know, but I'm also saying...
you make these sphingolipid out of your brickstones, and if you are low in some brickstones, and that could be serine.
Karsten, I know, and you're right, but the point is sometimes less is more in a trial, and the truth of the matter is, if this becomes known, I was 20 years ago, I tried to design a trial because I believe that metformin will help type 1 diabetes for a long time, right? Because it helps the function of insulin. It doesn't push the beta cells, it makes the glucose sensing better. The trial could not be done because all the reviewers said, but if people know about this, they will do lifestyle intervention. And lifestyle intervention is something you cannot control. And lifestyle intervention includes taking vitamin D, putting serine in your food, be easy on the gluten. And anything you do here is going to affect the power of your trial. That is the difficulty.
I wouldn't say that in Denmark, but here I think I could say it, because our trial is going on in Denmark. If you had one in US, I would not mention it, of course, because it is important.
But I would also disagree with that, Karsten, because patients have the right to know what's good for them. I don't want to hide anything for patients. That is.
Yes, of course, ideally, but as you said, as long as the trial is running, there's also a benefit for the patient that is going to be a fair trial.
Let me ask you guys this: if why not make a stronger, study by sort of testing for the levels of sphingolipids, maybe of the levels of serine, if you want to get that far out there. But
this
maybe
is in the islets, Monica.
Sorry?
This is in the islets.
I know, but is there any
mirrored.
marker? Is there a blood marker you could that might indicate?
Could be mirrored.
People are, this could be a subpopulation that would benefit more, and hand select those, do the short trial that Henrik suggested, a 50-day trial with a smaller number of people. Is that a possibility?
This can be really very dangerous as well, because if it doesn't give you a slam dunk result, people will turn away from this. So you have to be very, very careful about quick and dirty. Mm-hmm. And we have to be very careful about thinking about endpoints. So I am as excited and anxious as all of you that this is something that should be tested. I don't want to shoot myself in the foot, as has happened with vitamin D, because and gluten, people say, oh, gluten. No, that's because as soon as people get to read about this, the placebos are taking less gluten and more vitamin D. So this is great and good for them. But it means that it's much more difficult to get significance between the arms. What we are doing in the TOL-DC trial, all the patients get a super dose of vitamin D. So you tell them everything you have in your brain, including sulfatide, that they do the very best to give their beta cells a break, and then you have a good trial. But the point is, we need to also be creative in endpoints, and FDA does not accept time in range. It's Henrik, are you still online? Because you would agree that would be a fantastic outcome, right?
Yeah.
We have to be very, very careful that we do slow steps. Karsten is doing that. Karsten took the highest probability group to measure it. So it's the easiest to measure is in the group that Karsten is testing. Give Karsten a break. Let him do the interim analysis in September. see what we have in terms of glycemic control. Karsten, I'm sure you have all the secondary endpoints in there, right?
Yes, we have.
And most of these patients in Denmark will be on pumps and sensors, so they have the best glycemic control anyway. That's a problem. If not everybody is getting the best care, it's also more difficult to get significance. So let's see. It's a few months away. I'm actually thinking that we should think if Karsten is able to confirm safety in this group, and let's say that efficacy was not immediately there, that means that the FDA it's actually not a new drug, right?
No,
So maybe we don't need the FDA anymore, and we can just say, listen, we care about patients, and we don't, and if we can avoid that the FDA is dictating area under the curve stimulated C-peptide, which is a horrible test anyway. I think it's an unethical test, to be quite honest. But because beta cells don't like it either, trust me.
Yeah.
So I would take time in range, hypo, less hypos, anything. By the way, we discovered a new HbA1c, let's put it that way, at City of Hope just now, and we are exploring that. It's an extremely exciting new development, but it's one that is much more stable, and you can test it in urine, and it's completely out of the box, a fantastic small molecule, cannot be broken down. It's terrific. So if that can give you an indication of better glycemic control, because it's a marker of beta cell happiness, that would be something that you could use as an endpoint.
yes.
And as long as we don't have to do the FDA dance, we have a chance to be original and move very fast, accelerate the process.
Yeah. Yeah. No, that's interesting. When is that a paper coming out? I'm kind of curious about that.
No, it is not a paper yet, but we did test it in teddy samples, and we could predict in people that have antibodies who converts to diabetes and who doesn't. So that is pretty amazing. Not black and white, but pretty cool. But now we are working with Annette Ziegler in her fantastic population-wide study. I should also do credit to the person that did this. It's Sarah Schuck. She's magic. She came from Vanderbilt. She is a chemist, and she doesn't do immunology. This is not an immune correlate, right? This is a beta cell correlate. I am so super excited, and all I'm trying to do is make her life, her career, go faster.
Thank you so very much. I will stop recording. Thank you so very much, Karsten, for this presentation and for everyone's input and discussion. It was really fabulous. And I'm just going to stop recording, and thank you again, Karsten.
Yeah, I thank you also for the opportunity, and it has been a pleasure to be here.